I have the distinct honor of welcoming you to our Alan Edwards public lecture series that focuses on improving behavior through brain science. Tonight's lectures focus on neuroscience of sight restoration, with a talk by Professor Geoff Boynton and a conversation and talk with sight recovery patient Michael May. Before introducing this evening's speakers, let me just make a few comments about this lecture series and how it came about. This annual series is the result of the generous support of Professor Alan L. Edwards, who made a substantial gift to an endowment that ensures that this series can take place free of charge to all of us. So Professor Edwards was part of our department for half a century until his death in 1994. He was an outstanding teacher, researcher, and writer whose scholarship addressed personality measurement, statistics, and research design. And his statistics books were gold standards for psychological research. So the Edwards family contribution to the psychology department is an example of what can be accomplished with the support from members of our community. As outlined in the lecture show that I hope you enjoyed as this went through this evening, our Improving Behavior Through Brain Science series is one of our department's new visions that help raise our impact in society to a whole new level. Here, I'm going to ask that you join our efforts by becoming a friend of the psychology department by visiting our website, or you could speak with me or our department chair, Sherry Mizamori, after tonight's lecture. And I thank many of you in the audience who have already made contributions to our department. Without further delay, I want to introduce our first speaker. I'm delighted to introduce Dr. Geoff Boynton as our speaker this evening. Dr. Boynton earned his Ph.D. in psychology from the University of California at Santa Barbara. He's currently a professor of psychology in our department, and he joined us in 2007 after a decade at the Salk Institute in San Diego. His primary research interests are in neuroscience, and more specifically in sensory and perceptual processes. His research explores how attention shapes the way the brain processes visual information in our environment. He's pioneered the use of functional magnetic resonance imaging technology as a tool for studying vision perception, and his work has been foundational in shaping the development of vision science. His work has relevance in understanding applications as diverse as sight restoration, which we'll learn about today. And one of my personal favorites is how our attention operates so we can prevent ourselves by being hit by looming objects in our visual field that might be going straight at our head. He's authored many papers on these topics. He's been awarded grants from the National Institute of Health as well as private foundations. He serves on the editorial board for the Journal of Vision and Vision Research. Here at the University of Washington, Geoff is a member of the Center for Brain Imaging Directors Committee. He's also a highly regarded teacher of statistics and neuroscience in our department, and it's such an honor to introduce my colleague, Geoff Boynton, to talk with you this evening. Thank you, Geoff.
Thank you, Cheryl. I'm not going to talk about objects flying at people's heads today. Instead, I'll be talking about some of the research that I've been doing with my close colleagues, Ione Fine, Michael Baylor, and Errol Rokem, on work we've been doing trying to understand what the visual experience might be like for someone who has had surgery for a retinal prosthetic. And after my talk, which is going to be about 45 minutes, there will be some time for question and answer, and then we can do a transition over to our second speaker, who you can think of me as being in the warm-up band for Mike May, who we're lucky to have here tonight, who, in addition to having a very interesting life, is a sight recovery patient. And so he's going to tell you a lot about his experience of having his vision restored as an adult. So the work I'm going to be talking about is work related to something called retinal prosthetics. If you want to learn more about that and you want to do a web search, you might want to search for under the term bionic eye. Now, that's the colloquial term for this sort of technology. Now, when I think of, I'm a guy of a particular generation, when I think of bionic eye, I immediately think of Steve Austin, the $6 Million Man, the TV show from the 70s. Do you remember this? So back then, this is like, I think, 1974, I think, when the first season came out. This is the main character had, after a horrible test plane accident, had his legs replaced, his right arm replaced, and his left eye replaced with a bionic eye. And the whole idea was to make him, better than he was, faster than he was, and stronger than he was. Well, what we're doing, now that it's more than 40 years later, I would say companies, are actually developing such devices to try to restore vision in the blind. And we're really trying to assess how well these devices work to help the engineers develop these devices in the future. Full disclosure here, I am a Ph.D., not an M.D., so I think of this as the less useful kind of doctor. So my background is in math and computer science before moving and seeing the light, so to speak, and moving over to psychology and neuroscience. So if there's any kind of medical emergency tonight, I'm not much use, but if anyone has like a serious, urgent math or statistics problem, I can be like the first responder. So why all this interest in retinal prosthetics, and why is it so much in the news? Well, blindness is a big problem, and not just being blind is a problem, but the prevalence of blindness is a problem too. The most common causes of blindness in adult are disease-related, and the two main causes are macular degeneration and retinitis pigmentosa. Both of these are diseases of the retina, which is I'll talk about this in a moment in a crash course on the physiology of the retina. But these are diseases where the photoreceptors in the back of the eye, the light-gathering cells, degenerate. There are a variety of causes. Generally, the causes are genetic, and there are many, many genetic gene mutations that can lead to this sort of vision loss. And they're age-related, and as our population ages, the prevalence of age-related diseases is increasing. So this is a real problem. The two main classes of retinal degeneration, macular degeneration, typically leads to the loss of photoreceptors in the cones related to central vision, your foveal vision, and that tends to spread from the fovea to the periphery. Retinitis pigmentosa, sometimes called just RP, starts with degeneration of photoreceptors called the rods, which we use for night vision. So the first symptoms tend to be a loss of vision at low light levels. And that tends to lead to photoreceptor death in the cones, the day vision photoreceptors, and that tends to lead to loss of vision from the periphery to the fovea over time. I'm going to talk a little bit more about what happens in the retina for these diseases. But first, let's do a crash course on retinal physiology. So this is a kind of cartoon version of the eye and the retina. And as many of you know, the eye is much like a camera, at least the optics of the eye are, where the cornea and the lens help to form an image of the world on the back of the eye here. And the image is upside down and backwards. This seemed to cause a lot of trouble for the early philosophers. But if you think about it, the brain has always experienced the world this way. It doesn't know anything different, so there's really not a problem for the brain. It's not like we have to do this in order to see the world properly. In the back of the eye, there's a thin tissue, neural tissue, called the retina. And the tissue is the right name for it. If you ever see surgery for, say, retinal detachment, and you get to see what the retina actually looks like when it's removed from the back of the eye, it looks like wet tissue paper. It's thin, it's easily tearable, it's fairly delicate. If you zoom in on the retina, you can see that the retina actually consists of various cell layers. Light comes in from this end, and eventually, if a photon is lucky, it will hit one of these cells down here called photoreceptors. And these photoreceptors are amazing devices. They act like light waveguides that can collect the light. And if a photoreceptor does come in here, if it's lucky, it can do something called isomerize and lead through a really interesting cascade of events that effectively amplifies the energy in the photon, leading to an electrical change that can cause these intermediate neurons to start firing on its way to sending signals to the brain. These intermediate layers, amacrine cells, bipolar cells, horizontal cells, do some really interesting image processing. For the most part, they tend to add and subtract signals coming from the photoreceptors up in the bottom. And so, for example, this cell might respond very well to a bright light shining on that photoreceptor, but subtracting light coming from these photoreceptors. So early stages of visual processing happens in the retina. The signal that's leaving the eye is not like a pixel version of the world out there. And the information does leave the eye through this last layer called the ganglion cell layer. These ganglion cells are very specialized neurons that send spiking signals out the eye through basically a hole in the back of the eye, long cable all the way back into the middle of the brain, in a part of the brain, in the central brain called the lateral geniculate nucleus. It's interesting, you might have noticed here, that light comes down through here. It passes through all this junk that are not light receptive cells, right? And eventually, you hit the photoreceptors down here. This seems backwards, right? We are now, when you're staring at me, you're looking through a bunch of non-transparent cells in order to be able to see me. We can't see that, and we don't experience that, because anything that doesn't change over time in the visual system tends to fade away perceptually. And so why is that the case? Well, you read textbooks on this. There's a lot of post hoc explanations. Well, these photoreceptors down here are metabolically very active. They shed their so-called discs over time, and there's the vascular systems down here. Maybe there's something about keeping things clean and housekeeping. But actually, I guess last week there was some discussion about the octopus. Many of you were here last week. The octopus is back. It turns out that the octopus and other mollusks, their retina is flipped around the other way in a sensible way. The photoreceptors are right here. Light comes down, hits the photoreceptors. Then the signals pass through a layer of cells and out the eye. What's the deal? Why are we this way? Perhaps we're not as evolved as the octopus. I pointed this out to my father-in-law, who happens to be a philosophy professor at NYU, and he thought for a moment and said, maybe God is an octopus. So here's a painstakingly long drawn diagram from a well-known paper showing the so-called axon pathways of these ganglion cells and how they leave from a particular location in the retina, lead out to a particular part in your back of your retina, and then out the back of your eye to your brain. Now, this is an amazingly distinct pattern. It's like a fingerprint. And these ganglion cell pathways, well, they don't cross streams, because that would be bad. And if you think about it, you have to somehow get signals from every point on your retina out the back of the eye, okay? What's interesting about this is underneath the optic disc, because it's basically a hole in the back of your eye, there are no photoreceptors. So many of you might know that you actually have a blind spot for each one of your two eyes. And if you close one eye and hold your thumb out about arm's length, about 5 degrees off to the side, away from your nose, your thumb might disappear if you do it properly. Now, fortunately, we have two eyes, and fortunately, most of us have two eyes, and fortunately, the blind spot for the two eyes don't line up in the same place. So the other eye can take over, where one eye might have a blind spot. But it's fun to do, actually. I do this in faculty meetings sometimes, where I can stare straight ahead and make my faculty members disappear. I shouldn't say that, because many of them are here tonight. If I'm winking at you in faculty meetings, I'm not trying to communicate, it's because I'm trying to make you go away. In the fovea here, this is the foveal pit. This is where the center of vision happens. It turns out that the ganglion cells, here are kind of pulled away from the very center. So actually, in the very center, maybe your fingernail's width at arm's length, they're not looking through the ganglion cells. Light can actually hit straight down to the photoreceptors. So in the intact, healthy eye, this is another diagram that's very similar to the one I showed before, showing the different cell layers, photoreceptors, intermediate cells, and the ganglion cells. But in the diseased eye, for example, in late-stage retinitis pigmentosa, you can see what's happening here in this diagram is the photoreceptors have degenerated. And so they no longer can capture light and send signals to the brain, hence blindness. But interestingly, and most important for what we're talking about today, is that many of the remaining cells are still alive and potentially active. And so many of these sight restoration technologies are attempts to stimulate these remaining photoreceptors, non-photolight sensitive cells. And if you think about it, if you make a ganglion cell send a signal to the brain, the ganglion cell doesn't know that light made it go versus an electrical signal, so the brain might see a flash of light or something like that. And that's the basis of sight restoration technologies. There are three main classes of sight restoration technology. I'm going to focus on retinal electrical prosthetics, but I should talk about the other two first. These are newer and emerging technologies. We know less about their efficacy because there are fewer, none of these are actual, they're all in clinical trial stage rather than actual patients coming home with these devices. One is a very exciting idea, is called dealing with optogenetics. Optogenetics is the process of using a viral vector to effectively change the ion channel structures of cells that are not photoreceptive, like ganglion cells, and make them light sensitive, which is pretty amazing. So you can actually take a cell, like the ganglion cells that send cables that leave the eye, and make them sensitive to light. You think this would be like the magic bullet. This would cure blindness by simply turning parts of the eye that didn't used to respond to light, make them respond to light. That'd be great, but there are a variety of limitations for this. One of the biggest ones is that this technology, as it is right now, these light-sensitive cells, when they're made light-sensitive, are much less sensitive than a photoreceptor. And so you need 2 or 3 more orders of magnitude of light at least to make these things fire. So daylight light levels are not enough to make these work. So in order to use this technology, what these companies are going to have to do is put a camera pointing into the, a light source pointing into the eye, connected to a camera, which is producing a very intense light that make these things fire. So it's going to require some sort of prosthetic device in order to be able to make the brain see a signal. Another technology, which is really exciting, is gene therapy. You might have heard about some of this. Some of these are in clinical trials for like Leber's disease. And this is the idea of using viral vectors, usually like the rabies virus, to go and to basically inject it into the retina, the diseased retina, and either prevent further degeneration of the disease or maybe even allow photoreceptors to grow back again by literally altering the gene structure of cells in the retina, thereby effectively eliminating the disease. This, if it works, is a true cure for blindness, for these particular kinds of blindness. The drawbacks are, well, as I mentioned, there's many, many different genetic causes of these sight loss, and so that means you have to develop a different gene therapy for each particular type of sight loss. And also, it's not obvious if it's going to be permanent, but this is a very promising technology, but it's still in the early stages. The most developed stage of sight recovery are these so-called bionic eyes, which involve placing an electrode, an electrical stimulating device, on the retina, which, when you turn them on and send currents through them, will induce firing activity in the remaining cells in the retina, and therefore hopefully producing something that might resemble sight. And that's what I'm going to talk about for the rest of today. There are a large and growing number of groups that are exploring this technology throughout the world, and they range from full-fledged companies, like a company called Second Sight down in Sylmar, California. That's the company we're working most closely with, to just independent academic groups in universities, like the group in Melbourne, which are really just a bunch of professors and engineers that are trying to develop this. So some are for profit, some are just curious scientists, and it's a very large and growing group. For the most part, these electrical prosthetic devices are based strongly and heavily on cochlear implant technology. So cochlear implants are devices where you can send an electrode down inside the cochlea of the ear for people that have had hearing loss or born deaf. And with electrical stimulation, you can stimulate the remaining cells in the ear, thereby sending signals to the brain that resemble, that sound like things, right? Now, cochlear implant technology has been quite successful, especially when implanted at people at a young age. And so much of this technology has been developed to use in the retina. In fact, this company, Second Sight in Sylmar, its sister company is a company called Advanced Bionics, which is a successful cochlear implant company. And in fact, the first test implant that Second Sight used had 16 electrodes. It was a 4x4 grid of electrodes. And it was 16 because their successful cochlear implant at that time had 16 electrodes in a line right there. How does this work? Well, there's actually a variety of places in the retina that you can place one of these devices. Second Sight has their retinal implant placed right here on top of the ganglion cells, it's called an epiretinal location. Another potential location is the subretinal location, which has been the layer between the photoreceptors and the back of the eye. And of course, there are no photoreceptors in these diseased patients, so it's somewhere between the ganglion cells and back here. The advantage to epiretinal here is that these electrodes are very close to the ganglion cells, which we're trying to stimulate. The disadvantage is that there's nothing really to hold this electrode and keep it from moving around. And Second Sight actually has a patented tack system where it's like stapling this thing to the back of the eye. But it can still maybe lift off a little bit and move around. If you stick it in between layers of cells and other stuff, it can be wedged in there and not move around as much. The disadvantage of this is it's further away from the ganglion cells, and also there's an issue of heat dissipation. So changes in current can lead to heat, and because it's stuck inside a bunch of stuff, it's harder to have these things not heat up and burn out the electrodes, and even worse, burn out the retina. The last one is kind of interesting. This is a group of various groups, like the one we're talking with in Melbourne, where you can actually stick this electrode outside the eye, basically in the back of the eye, called the suprachoroidal location. And this makes surgery really easy. You don't need to make slits in the eyeball and stick this thing inside the eye. You just slide it around the back of the eye and tack it on back here. Makes surgery very easy, relatively easy, and also it makes it easy to replace as technology improves. You can just pull it out, put a new one in relatively easily, presumably. It has the disadvantage, of course, that we're farthest away from the ganglion cells as any of the other methods. And as the further you go, the bigger the spread of current you're going to have. And so the ability to stimulate a very localized part of the retina becomes more and more difficult. But what we've seen from the results from this group are remarkably good. I'm going to show kind of a cheesy movie pulled straight from Second Sight's website, because it's probably the best description of how this technology works. And so I'm going to take a drink of water while this thing runs and let this thing go. And watch the end of this movie, because there's something kind of important about the way they think people are going to use this.
In Second Sight's Argus II retinal prosthesis system, a miniature video camera in the eyeglasses captures the scene. The video is processed by a small portable unit and transformed into instructions which are sent back to the glasses. These instructions are then transmitted wirelessly to the implant on the eye. The implant consists of a receiver and an array of electrodes. Instructions are received and corresponding signals sent to the array, which emit small pulses of electricity. These pulses stimulate the retina's remaining cells and are transmitted down the optic nerve, conveying visual information to the brain, which perceives patterns of light. Patients learn to interpret these visual patterns.
Second Sight was the first company to develop these prosthetics and get FDA approval to do this. So they're on the order of dozens of patients now that have gone home with these devices. So there's lots of data to be gathered from them. I don't know if you noticed at the end of this video was the implication that if you are staring, so here's another image taken from their website. If a patient is staring at a doorway, so here's the region of the visual field that they have vision loss. If there's a doorway that's behind that, if you just simply stimulate the electrodes in such a way that resembles the shape of the doorway, the patient will see the shape of a door. They're a little careful in the wording there. They say patients learn to interpret the input, but they do have this picture right here. And in this picture is the implication that if I turn on an electrode right here, it's going to lead to the percept of a single spot of light at the corresponding location where the photoreceptors used to be underneath that electrode. And we call this the the scoreboard model of visual percepts. It's called the scoreboard model because if you've ever been to a stadium and walked up and looked up to one of those big stadium scoreboards really close, you'll notice that the scoreboard is actually made up of a bunch of individual light sources that can turn on and off at different intensities, different colors, and it looks like a continuous image because if you stand far enough back, the visual system doesn't have the spatial resolution to see the individual lights. And so with a high enough resolution, you can fool the visual system into seeing something that looks like a continuous image. And of course, in fact, this screen right here is the same principle. If I look really, really close, I can see individual pixels that you can't see because you're sitting too far away. So presumably, if you have enough electrodes, and each produces its own spot of light when I turn it on, With brute force advances in technology, all I need to do is increase the number of electrodes, and I should eventually be able to restore sight. So the first thing you want to know is, well, is the scoreboard model valid? Well, one way to test it, and this is something that my colleague Ione and other people at Second Sight have been doing, is to literally ask patients, what do you see when you turn an electrode on? And the way they can answer the question in this particular example is to have a patient draw with their finger on a touch screen right in front of them. It's pretty crude, but effective, a touch screen, what they perceive. And so, for example, if you were to have a 4x4 array like this, like the early versions of Second Sight's prosthetic, and turn these two electrodes on, ideally you should see 2 spots of light in those 2 locations. But what do they actually see? Well, this is an example of one patient's drawings. One of these electrodes led to a fairly nice punctate spot of light, but another one led to this really long, weird banana-shaped streak. These are different grayscales because what we've done is drawn on top of each other various replications of drawing over a bunch of trials. So it shows that her ability to draw is very easily replicable. So she sees this streak every time you turn this electrode on. Well, what's going on here? This is clearly a problem with the scoreboard model. Another problem is that over time, if you turn an electrode on, the percept fades. Some of you might know this about the visual system. If you stabilize an image on the retina, it tends to disappear over time. Something called Troxler fading. It's a big problem in the retinal prosthetic industry. Here's an image showing, drawn on top of, this is now an A60, this is the 60 electrode array. Drawn on top of that are streaks drawn by a patient for the one half of the electrodes and the other half of the electrodes. This is divided in half so these aren't all lying on top of each other. And you can see that there are very few spots and many streaks. Here's another patient showing also not spots, but thicker streaks, different percepts. Not quite sure why they're quite different, but it might have something to do with how far away this electrode is sitting from the ganglion cell layer, because it does vary from patient to patient. You can see there's some systematicity in the way these streaks are pointing. They're not all just random directions. And we think the cause of this has something to do with the way the ganglion cell pathways, axon pathways, are leaving the eye. So for example, suppose you're stimulating with an electrode right there and a patient sees a streak like that. Well, it turns out that underneath this streak, if you overlay or underlay this axonal pathway diagram, you might see something like this, where the streak is following these pathways. And if you think about it, if you stimulate right here, there might be a ganglion cell who started its pathway right there that was connected to photoreceptors originally underneath there, and so sure enough, you're going to see a spot of light in the right place. But also, suppose you have a ganglion cell that starts right here and so it represents this part of the visual field, but sends its signal along this pathway underneath the electrode and out the eye. If that's the case, stimulating here was also going to stimulate that axon wire, and thereby leading to a percept of light over here, and so on all the way down the pathway. So we believe in our modeling ideas this is what's causing these particular streaks. If you stick an electrode right here, you might not see a long streak because the pathways basically end right there. And so we've been able to match up these perceived streaks with this particular kind of diagram from patient to patient with reasonable success. Another issue, we've got lots and lots of data from these patients, measurements such as how bright does the percept look, regardless of its shape, as I change the current amplitude into the electrode. And what you'll see is a typical nonlinear relationship, where as I turn the amplitude up, I don't get this linear increase in perceived brightness. And this is a simple experiment where subjects are simply giving us a number from 1 to 20, or 0 to 20, how bright this thing looks to them. There's a whole bunch of other so-called psychophysical measures using brightness matching techniques, threshold detection techniques, which is like how much current do I need to be able to see a flash of light? Huge piles of data coming from Second Sight and other companies that we're trying to compile to try to understand what these patients are seeing. And ultimately, what we're trying to develop and are developing is something that we call a virtual patient. So given some pattern of electrical stimulation on the retina, a patient might see some kind of percept. Our goal is to take the same input, patterns of stimulation along, say, 60 electrodes over time, pass it through a model that we've been developing to create, and the output of the model is going to be basically a movie that simulates what this patient will see. And so for this particular example, our model output for stimulating these two electrodes gives us that right there, which is a reasonable match with what the percept of the subject or the patient was seeing in this example. Now this model is based loosely on retinal physiology, these various stages of spatial and temporal processing, but the parameters of the model have been constrained by the data sets that we get from the behavioral data. So it's kind of a nice hybrid between a physiological model that's constrained by data, behavioral data. And so how is this coming along? Well, one of the main reasons why we're doing this, some obvious reasons. One of them is it provides a more realistic estimate of how visual prosthetics are likely to perform. And this is useful for doctors that might be implanting these techniques to be able to tell the patients how well these things might work. It's great for engineers, so they get an idea of how well these things are going to work before they actually build it. And it can provide insights into what sort of visual tests are important for evaluating the devices. You'll see in a second that it's going to be pretty obvious that these devices are not going to be very good for things like facial recognition. And so there's no point in trying to do a facial recognition test on these patients. And finally, the interesting part is we're going to try to invert this model so we can find the best pattern of stimulation to lead to a desired percept. So we call this a forward model, where given some input, we have some output that is going to be some movie over time. But what we really want is, suppose we want to have the letter A over here at the end, what pattern of stimulation do I need to produce that output? That's actually the model working backwards. And because this model is so complicated and nonlinear and slow, it's going to require some pretty tricky techniques to be able to do this. And we're actually collaborating with the groups at eScience in the physics building to use some of these deep convolution learning algorithms to basically train up this network, given an input and a desired output, to figure out how to make this thing work backwards in just sort of a brute force manner. So how well is this working right now? Well, here we go. Can anybody guess? This is our best guess of what a patient wearing the Argus 2, which is the 60 electrode array, what they might see given a very large letter presented to them. Anybody guess what that is? Very good. It's a black A on a white background. How about this one? It's actually also an A, but it's actually a little easier to see, probably, to many of you. And that's a white A in a black background. And this is something we've actually learned from our simulations, that it seems to be easier, at least for us sighted people looking at the output of the model, to recognize a letter when it's white on black versus black on white. And indeed, it turns out that Second Sight, when they send these patients home with these devices, they have a series of knobs on their computer that translates the camera image into the stimulation pattern. And one of the knobs allows them to do contrast reversing, which basically takes a white part of the visual field and turns it into low-level stimulation, and a dark part of the visual field into bright, high levels of visual stimulation. And many of these patients are said to prefer this contrast reversing mode rather than the standard mode where dark parts of the field are low modes of stimulation. It's kind of interesting, and it's almost a philosophical point here, where are we trying to restore sight, as in how we see, or are we trying to restore functionality? So you and I who are sighted would never want to reverse the contrast of the world to function better. But for some reason, because of all the weirdness of this distortions of the image, it actually is easier for patients to be able to use this device when flipped around and contrast reversed. So this is restoring function, but not necessarily restoring traditional sight. I think this is a really important distinction we try to make when we're talking about this work, especially with the engineers. We can also make movies of this. So here's a stock movie of, I think this is the Boston subway of people walking around. See if this works. There's no sound here, which is good. You can see there's a person walking from left to right, and there's some motion right to left, and the car starts moving here. This is what we have, our best guess of what a patient might see while looking at this movie. I'll show this a couple times. And what I see is the basic motion of a person moving from left to right. I don't see a lot of the aftermath of people moving right to left in the distance. and you can be a judge of whether this would be a useful device for somebody that's blind. I would say that overall it's better than nothing, right? You can see whether there's global motion going on. And in fact, these patients say that these devices are useful for major global motion and not so much for fine-grained detection of movement. I've got a few more examples. This is a fun one. This is in England, northern England, where we spent a year. This is my nephew schooling his older brother in an epic soccer goal right here. Boom. His older brother's not too pleased about that. I probably shouldn't be showing this without his permission now that I think about it. Because he didn't want this movie in a publication that we made. Oh well, just pretend you never saw this. What happens here stays here. But this is a little disappointing. This is less of an epic soccer goal as seen through the prosthetic device. Not a whole lot going on here. And part of what you notice is when I filmed this in the backyard in England, I track the motion. I actually track the camera partly with the cousin, so there actually isn't much camera motion on the image itself. So there's not much to see, except until the end. This movie's in black and white. Some people might ask about color. Restoring color vision with these prosthetic devices is, at best, a very long way off. If you think about trying to excite these ganglion cells to send a color signal to the brain, we hardly understand how ganglion cells send color signals to the brain, so we're way, way off from that. So this movie was originally filmed on my iPhone in color, but it was northern England in the winter, so it didn't really matter anyway. Here's another one, northern England, of my then 7-year-old practicing for the X Games on a scooter. This is a good one because it really has nice, clear right to left motion, and it's pretty easy to see that in the movie. You wouldn't know that this is a kid on a scooter, but it's definitely something moving right to left, and you might want to avoid it if it's, making a lot of noise, right? So, finishing up here, where are we with electrical prosthetic technology? Well, Ione has a really nice analogy where we like to think of where we are with electrical prosthetics as sort of where human flight was maybe the beginning of the last century. So back then, people were building kind of ridiculous looking things and strapping it to their back and jumping off cliffs and trying to see if these things worked. And there's a lot of trial and error, and some things worked, and if they did, they adapted them and moved on, and eventually we are where we are now. But it was a lot of trial and error. Where are we now with flight? Well, here we are in Seattle. The Boeing 787 was developed almost entirely with computer-aided design and simulations for mechanical engineering and aerodynamics and heat dissipation. And by the time the first 787 was built, they pretty much had a good idea of its flight characteristics, and there weren't a whole lot of surprises. Except, I think they were worried about the wings falling off or something like that. But aside from that, the idea is that if you know enough about how the system works, you should be able to design something and build it and have an idea how well it's going to work before you actually try it out. And what we're doing with our group with these simulations is try to get retinal prosthetics, help them get from here to there, so that an engineer will know what size electrodes, the array of electrodes, what pattern of stimulation, what's going to work best before you even build this thing and stick it in somebody's eye. And as you know, as I pointed out, these visual percepts are not ideal. They're quite distorted. And if you read articles on this sort of thing from these groups, typically in the discussion section of these articles, there's some talk about, well, the visual system in the human brain is very plastic. We can learn stuff. And so perhaps patients will learn how to use this complicated and distorted visual input. And that may be true. We learn a lot. You learned something about the octopus eye today, and you'll probably know that for a long time. Your brain has changed because you listened to somebody tell you something. Well, the visual system can learn too. And as you know, if you meet somebody new, you've seen a new face, you can recognize that face later on. Something in your brain has changed when you saw that face. So neural plasticity in learning can be very helpful. On the other hand, there are limitations to learning. And this is something we're going to talk about and learn about in the second half of tonight, where Mike May is going to come up after a discussion, or question and answer, and he's going to talk about his experience of having his sight restored as an adult and the successes and maybe lack of success that he's had on using this brand new sensory input. So thank you, and if that's, I think there's a microphone back there.
Okay, my name's Angie, and my question is with two-part. First one is with the surgery for the microchip to be embedded into behind the eye, the optic of the eye in the retina. Is that invasive, laser, or is it actually going to involve a scalpel to? And then second question is with the neurological system so close to the eye orbit, would it interfere at all with the brain transmitters that we have with our, like, say, our endorphins and our natural sensors? I mean, kind of like a short circuit type of thing because of it's an actual metal inside the eye.
Yeah, I can see what you're saying. Well, the first question is, I think you're talking about the suprachoroidal technique, which you're embedding this prosthetic behind the eye. That doesn't require any opening up of the eye or heavy surgery like that, as far as I know. So it's relatively easy surgery. You just go around the outside, underneath this, around the socket of the eye. The second question is whether these electrical signals that you're sending into the retina can interfere with other kind of neural activity, I think is what you're asking. And the efficacy of these electrical currents that you're setting up that drive these cells really drops off quickly. It's an inverse square law with distance from the neurons. So I seriously doubt that an electrical signal in your retina is going to affect any brain activity right behind that, say, which is important because your frontal lobe is right behind your eyeball, right, and your supraorbital cortex and all that. So I don't think that is the problem. But it's an interesting question, yeah. Yeah, thanks. Go ahead.
Is any of this applicable to a detached retina?
I don't believe at the moment. So detached retina is when basically all the layers come off from one part, and it requires some surgery to basically correct the retina, put it back down on top of the sclera. As far as I know, there would be no need to, because the retina still functions, it's just in the wrong place. So I don't think it makes any sense to stick an electrode on top of a functioning retina, even if it is somehow moved or lifted off the back of the eye. Yeah, interesting question. Yes?
Yeah. What are the major roadblocks that you see between you and where you want to be? Like, is it the electrodes? Is it electronics, the processing, the data?
Great question. Yeah, what are the major roadblocks? So I think it's clearly the case that exciting a given part of the retina with a small electrode is going to lead to something related to the scoreboard model. So the streakiness that we're seeing is a big problem. This fading over time is a big problem because you can't just keep turning up the current level to compensate for the fading, because eventually you run into current level problems where you overstimulate and can cause heating of the retina. But I think a lot of these problems can be compensated with what we're doing, is coming up with more clever techniques of stimulating patterns to try to produce a desired output, rather than just focusing on simply a relationship between, say, the brighter part of the camera image is going to relate to a higher current for that particular electrode. But I think the major limitations are the spatial blurring and the temporal fading that we're talking about.